Hello students, welcome to chapter 7. We're going to be talking about cellular respiration and the harvesting of chemical energy through sugars and, starches and oils and fats and proteins and things like that. Okay, basically carbohydrates. So where we do this is in mitochondria. So mitochondria forms ATP as a part of the reactions of cellular respiration. So cellular respiration itself is the collection of metabolic reactions that break down those glucose molecules, break down food molecules that produce energy in the form of ATP. So the overview of cellular energy and metabolism. Cellular respiration does this by breaking down glucose or breaking down any type of glucose type molecule. So they contain electrons at a high energy level because they have those carbon molecules bound together, carbon and hydrogen molecules bound together. They're major biological steps of the carbon cycle. So that's one step, and then photosynthesis is the other step. Photosynthesis stores energy from sunlight in the energy-rich fuel molecules such as stored as sugars. So it converts whatever cellular respiration breaks apart into sugars. And then cellular respiration moves the sugars back into CO2 and water. So it basically hydrolyzes that glucose into CO2 and water. So the two reactions that are taking place here are oxidation and reduction reactions, right? So oxidation, we have the removal of electrons from a substance. Whenever you remove electrons, that substance is oxidized, right? Whenever you add electrons to a substance, that's reduced. And it's a little counterintuitive than what you might think. Whenever you add something, you reduce it, and it's not, it's not what you think of, right? But if you think about it this way, oil rig. Oxidized is losing, reduced is gaining, right? Oil rig. So oxidized is losing electrons, reduced is gaining electrons. They're always in coupling reactions, right? So it's always one is oxidized and one is reduced, right? The electrons have to go somewhere, right? So one is oxidized, one is reduced. So we call them redox reactions. We reduction, ox meaning oxidized. So redox reactions. We have electron carriers. These are the things that are going to be either reduced or oxidized. So dehydrogenase enzymes facilitate transfer of electrons from a fuel molecule to a molecule that acts as an electron carrier. So we have nicotinamide, adenine dinucleotide, or you can just call it NAD+, right? It's plus because it usually contains some other ion that doesn't need to be introduced. And then when it's reduced, right, NAD plus is in an oxidized form. When it's reduced, it contains an extra hydrogen molecule, right? It'll be reduced, it'll add on an extra hydrogen molecule, right? And then it'll do its job. Cellular respiration dehydrogenases transfer 2 electrons and 1 proton. So the proton is the hydrogen, right? So resulting in complete reduction to NADH. That's its reduced form. So the proton there attaches, completing it to NADH. So the first step in cellular respiration is glycolysis. Enzymes break a 6-carbon molecule into 2 3-carbon, what's called pyruvate. And pyruvate isn't specifically called triose because pyruvate adds on other things to it. So first we have a glucose, ATP comes in, adds a phosphate to it. Then we have glucose 6-phosphate, right? And then it changes to fructose 6-phosphate, right? That's a ketone. Remember, it changes the carbon-oxygen bond. And then ATP adds another phosphate to it. So it's fructose 1,6-diphosphate, right? This is called substrate level phosphorylation. Anytime you add a phosphate group to a substrate by ADP, by ATP, this is called phosphorylation. Adding another phosphate to a group, that's phosphorylation. Okay. Adding that, it binds it together, it mixes it up, it makes 2-glyceraldehyde 3-phosphate. Suddenly the NAD comes through, it takes off 1 proton. And then another NAD comes through, takes off another proton. So that's 2 NADs that come through, taking off 2 protons from that glyceraldehyde. So you have 2 glyceraldehyde 3-phosphate, right? Because that fructose split apart, making glyceraldehyde 3-phosphate. And then you have 2 1,3-diphosphoglyceraldehyde. 2 ATP come in to those 2 molecules, making 2-phosphoglycerate, right? So the glucose has been split apart by now. Some other things go through, and then you have 2 pyruvate, right? You have 2 pyruvate by now. All right, all of this is happening outside of mitochondria. All right? The reason why this is happening outside of mitochondria is because glucose is much too large to fit inside the mitochondria at its level. So if the glucose is this big, it can't fit inside the pores going inside the mitochondria. It has to break apart into pyruvate, and then it can go inside the mitochondria. You can also convert that into lactate. You can convert 2 pyruvate into lactate, but it doesn't go into the mitochondria, it just stays outside. Okay, after it converts into pyruvate, it can go into the mitochondrial matrix. Enzymes convert the 3-carbon pyruvate into 2-carbon acetyl group. Okay, it basically adds a co-acetyl enzyme to that pyruvate and completely oxidized into carbon dioxide. Okay, some ATP is synthesized during the citric acid cycle, all right, but once that goes into the citric acid cycle, it needs to remain, that CO, that acetyl CoA, it needs to remain on that chain. Okay? So then you have a pyruvate dehydrogenase complex. NAD comes through, adds an H onto it, adds that proton onto it. CoA attaches onto that pyruvate. And then you have this acetyl-CoA complex. And that's important in the citric acid cycle. All right, so then we get into the citric acid cycle. Carbon products of pyruvate oxidation, or the acetyl-CoA, cycle, are oxidized into CO2. We just talked about that. All available electrons are transferred to 3 NAD+. Okay. And then 1 FADH. So 1 FADH goes off and gets another proton, FADH2. Each turn of the citric acid cycle produces 1 ATP by substrate level phosphorylation. So you have this big long cycle, and I'll show you that in the next slide. Every cycle of that produces 1 ATP. The 8 reactions of the citric acid cycle, or tricarboxylic cycle, or Krebs cycle, whatever you want to call it, oxidize acetyl acid groups completely into CO2. Okay, so whenever you're breathing out, that's all the CO2 that's coming from your mitochondria. This is generating 3 NADH and 1 FADH2. Okay, all of those electrons are being carried away by those electron carriers, right? And every cycle, it produces 1 ATP. So at the bottom here, we have the equation for the citric acid cycle. So we have 1 acetyl-CoA, 3 NAD plus 1 FADH plus 1 NADP plus 1 phosphorus plus 2 H2O, 2 water molecules. That comes into the citric acid cycle. And then that gives 2 CO2s, 3 NADHs, 1 FADH2 plus 1 ATP per round, plus 3 hydrogen molecules plus 1 CoA, and that comes back up to the top to give back the acetyl-CoAs. Okay, so this is the whole cycle coming around from pyruvate giving the acetyl-CoA coming into the citric acid cycle. So you can see all the different enzymes going through the system, and the water molecules going in and out of the system, and then all the CO2s coming in and out. So without all of these enzymes and everything, if one of those enzymes were to just disappear, that citric acid cycle would stop. So the next step in this cycle comes into the inner mitochondrial membrane. Okay, so we were in the matrix, basically inside that center of the mitochondria. Now we're inside the membrane, in that what's called a cristae, basically those folds in the membrane of the mitochondria. Okay, and this is called oxidative phosphorylation, also called the electron transfer chain or electron transport system. This whole system goes through different cycles of those membrane proteins. So you have electron transport proteins, you have 1, 2, 3, 4, and what's called ATP synthase. Free energy released by electron flow generates a gradient of hydrogen molecules. Okay? And this gradient of hydrogen molecules goes through and goes to that ATP synthase. And it uses that gradient as an energy source to make ATP. And this is where the greatest amount of ATP is made in the mitochondria. So we have 3 major protein complexes numbered 1, 3, and 4. There's a smaller complex known as complex 2, and this is bound to the inner mitochondrial membrane. And all of these hydrogen molecules are brought by those electron transport molecules, right? So NADH and FADH2, it brings those hydrogen molecules up to those protein complexes and just kind of drops them off. It pulls it through those transport protein complexes, and it puts them into the inner membrane space between the first and the second membranes, between the cristae, right, and the outer membrane of the mitochondria. There's a proton gradient of those. Okay? So there's a high concentration of protons on the outside. There's a low concentration of protons on the inside of the mitochondrial matrix. So what does it want to do? It wants to diffuse through that membrane, right? So when it wants to diffuse through that membrane, it goes through the ATP synthase. So if it wants to diffuse through that membrane, for your attendance assignment, what is that diffusion called? Okay? What is the diffusion called of hydrogen molecules through the ATP synthase? And this is in your book, you can look it up online. What is the diffusion of hydrogen molecules through ATP synthase, or basically the diffusion of hydrogen molecules? So those cytochromes, protein with heme prosthetic group that contains an iron atom, right? Heme just means iron. So the ATP synthase gathers all those hydrogen molecules, and then it starts spinning like a windmill, right? ADP comes through with a phosphate group. Once that phosphate group latches onto the ADP, it becomes ATP. And every single hydrogen molecule that comes through there latches on that ADP with that phosphate group and becomes ATP. So every single one catches an ATP. So you can imagine every spin is an ATP molecule. That generates a lot of ATP. So overall, we have cellular respiration. It releases energy from electrons as they pass among acceptor molecules to the final acceptor molecule, which is oxygen. Okay, so the final acceptor molecule is oxygen, and that converts into CO2. Total energy obtained but depends on the difference between the high energy level in the fuel, right? So it depends on if it's glucose, if it's maltose, if it's whatever that you ingest. So the lower energy level in the final acceptor, right? Whatever oxygen level that you get. Cellular respiration includes reactions that transfer electrons from organic molecules such as glucose to oxygen, and reactions that make ATP. Okay, cellular respiration includes all the reactions from glucose to oxygen and reactions that make the ATP, reactions that make oxidative phosphorylation. Anything from glycolysis to oxidative phosphorylation. So overall, we have the glucose, C6H12O6. We have oxygen, ADP plus phosphate. Water turns to water, CO2, and ATP. And 32 is a round number. Okay, it can go from 32 to about 36, depending on what the energy level is in your glucose molecule. Okay, so we have two different types of respiration. We have aerobic respiration, where you inhale oxygen, right? That's what we do. So aerobic respiration requires oxygen. So eukaryotes require aerobic respiration. Many prokaryotes do as well. This is what mitochondria require. Oxygen is a reactant in the ATP producing process. But then there's anaerobic respiration. So anaerobic respiration uses a molecule other than oxygen, such as sulfates or nitrates. So it gets their oxygen, so to speak, from sulfates or nitrates or other types of things that have oxygen in the compound for its ATP producing process. And then there's fermentation. So when oxygen is absent or limited, certain organisms will go through just fermentation. Okay, so these electrons carried by those electron carriers, NADH, such as NADH, will produced by glycolysis, may be used by fermentation. So they're transferred by an organic acceptor molecule, converts that NADH to NAD+. They accept the electrons in reaction 6 of glycolysis, and that continues to supply ATP by that substrate level phosphorylation. All right, so the difference between fermentation and anaerobic respiration differs in the Krebs cycle, right? So anaerobic respiration still has the Krebs cycle. And you can see it right here. So the anaerobic respiration transferred to an electron transfer system in which the final electron acceptor is a molecule other than oxygen, such as the sulfates. In fermentation, electrons carried by NADH are transferred to an organic acceptor molecule. Right? It transfers to alcohols or acids or whatever. So anaerobic respiration can get either 2 or 36 ATP. Fermentation only gets 2 ATP, depending on where it goes. in fermentation, there's two types. We have alcoholic fermentation, we have lactate fermentation. So alcoholic fermentation converts the pyruvate into ethyl alcohol. This converts that also into CO2, right? So if you convert that into ethyl alcohol, it's also going to emit some CO2 as well. This happens in plant tissues, invertebrates, bacteria, and also yeast. So if you're making bread or beer or wine, it's going to be fizzy in your beer, and then the bread is going to expand, right? You're going to see those bubbles in the bread. So all that is CO2. In lactate fermentation, this converts the pyruvate into lactate, so lactic acid. So this occurs in bacteria and plant tissues. This doesn't produce a whole lot of CO2. So this is used to make buttermilk or yogurt or dill pickles. So it's an acid, it's going to taste very acidic, it's going to taste sour. There are certain species of bacteria, there's certain species of fungus that only go through fermentation. There's also certain ones that will switch between fermentation and oxidative pathways. So strict anaerobes are those that only go through fermentation. So the first one is Clostridium botulinum. That's one that causes botulism. And then Clostridium tetani, that's one that causes tetanus. They're both in the same genera. So they both will, if they get into your skin and the skin closes off, it will cause those symptoms, those diseases. And then facultative anaerobes, they can switch between fermentation and oxidative pathways. So things like yogurt. There's certain bacteria inside yogurt that will, ferment, and then they'll give you that type of acid, that lactic acid that you can taste. So there's also different types of glycolysis, and there's aerobic glycolysis, and then there's called abnormal glycolysis. So aerobic glycolysis is a process of higher than normal rates of glycolysis that can occur in cancer cells. This is something that happens, this is something that's called a Wahlberg effect. So you have one glucose molecule and then two lactic acid molecules and NAD, and it picks up hydrogen from two NAD molecules, picks up hydrogen from glucose, and it goes through glycolysis and then gives it to the lactic acid, and it goes through fermentation. And that basically gives it the ability to go through this whole big cycle of fermentation through these cancer cells. it generates a large amount of lactic acid, and it's not very good for inside the system. Because if you know this, if you work out a lot, that burning that you feel inside your muscles is lactic acid. If you're not breathing a lot, the oxygen doesn't get to your muscles. It's actually anaerobically causing that lactic acid to build up. That's why you need to drink a lot of water. That's why you need to breathe whenever you're working out, right? So if you have a large amount of lactic acid building up around your cells, it burns. Oxidation of carbohydrates, fats, and proteins. So many of these carbohydrates, fats, and proteins can be hydrolyzed. So if you have a large protein or large carbohydrate, they can be broken down, and their products can go in directed into various stages of cellular respiration. They can be oxidized as fuel. Fats, large fats, produce twice the amount of energy of oxidation of proteins or carbohydrates. That's why when we were in the earlier stages of development as humans, we would eat a lot of fat tissue, because that would give us the ability to go running, that would give us the ability to go jogging and such, to go hunt, especially if it's cooked. Cooked fats have a better ability to go through our system faster and have the ability to go through our system at a much easier rate than uncooked fats. So before entering oxidative reactions, triglycerides are hydrolyzed into glycerol and individual fatty acids. So as soon as those fatty acids are broken apart, we can hydrolyze those faster and give our bodies the energy it needs in order to do all the ATP synthesis to get our bodies moving. Proteins. The amino acid group is removed. Once that amino acid group is removed, that can be broken down into carbohydrates and then put into our system as pyruvate and glucose and whatever it needs to be. Make sure to get that attendance assignment done, and today is your lab practical, so get that done as soon as you can. It's going to stay up until tomorrow. Okay, so get that done. Other than that, have a good day, and I will see you later.